Hi, everyone. This is a short video on meiosis. We had just ended talking about mitosis, which was essentially to produce all your cells in your body except your sex cells. So here we are with meiosis, which is now really strictly for sexual reproduction. So meiosis, unlike mitosis, they divide twice, whereas mitosis, the cells were dividing once. In meiosis, you are creating four daughter cells. Remember, so we are dividing twice. This is why we end up with four daughter cells at the end of the cell division. And those four daughter cells are essentially those gametes or sex cells that you know of. The gametes are genetically different because they have half of the chromosomes that they originally had in the beginning. So each daughter cell contains half, as I said, as many chromosomes as the original parent cell. So if the original parent cell had 46 chromosomes, then that would mean after two divisions, the daughter cells would have half of the chromosomes, which is 23 in the case of a human. If, let's say, an animal, I believe like a horse, if the parent cell had 200 chromosomes, then the daughter cells would have 100 chromosomes. So in meiosis, the end result gives you cells that contain half the information, which is why they are called haploid cells, as in half. In mitosis, the cells, the daughter cells, are diploid cells, double. But in this situation, the sperms or the ovums only contain half the chromosomes. The purpose of meiosis is for male and female gametes, as you know, to be produced in order for reproduction. That is really the only purpose. And it is done so in the reproductive organs. So the ovaries for women and the testicles for men. And so that's it. We result with four haploid daughter cells. This is an image that you may run into sometimes, simplified. What you need to pay attention to are the parent cells and the daughter cells at the end. Pay attention that in mitosis, the division happens only once, whereas in meiosis, the division happens twice. One time here, one time here. This little animation, meiosis is very well portrayed. So as you see, this is the first beginning of the first division, the prophase, where they get all condensed chromosomes, and we create two pairs of homologous, which means identical. Then they line up in the middle, as we had said before, for metaphase, and they stretch out and they move away from each other, which is anaphase, and they finally recreate a new nuclear membrane into telophase. And lastly, cytokinesis, we're going to have two cells. Then for mitosis, that would have ended there. But now we're at meiosis, so we're continuing to division. And all of it starts over again, as you can see. And the only difference is there's no duplication. Instead, there's a crossing over. It's a mix-up of chromosomes. They're all different, as you notice here. And we end up with four different haploid cells. The parent cell in the beginning, let's see how many we had, how many chromosomes. There were four, or 8 or 4 pairs, if you want to say. And then we end up, look at that, each one has only 2. We started with 4, we end up with 2. And in a microscopic world, let's take a look. We have here an example of a meiosis under a microscope. So as you can see here, we are slowly starting to see the anaphase and now telophase over here. It's a little quick, but you can tell that it starts with now 2 and then 4. Look at that, how they divide it. And they will divide again. This shows you how slowly but surely we end up with 2 separate cells eventually, 4 separate cells eventually. Look at that. 1, 2, 3, 4. One, two, three, four. There you go. And that's it for me.